Hi everyone, so following up from last time, we put our normal cells called CCD841 colon cells from a smaller flask into a larger flask. So now the cells have filled the flask. We'll call this confluent, and we're going to have a look under the microscope. So now that we've had a look at the cells, what we're going to do is discard of this old media. You can see it's turning orange. And then we're going to rinse it with some phosphate buffer. Then we're going to discard that. Then we're going to use trypsin to lift the cells off the flask and count how many cells we need so that we can put them into 96 well plates. Okay? And now we're adding PBS, 5 mils. And we gently rock over the cells, discard. And finally, we add trypsin, 5 mil as well. And we pop this into the incubator for about 5 to 10 minutes until the cells have lifted. Okay, so it's been about 10 minutes, and we're gonna have a look at the cells, and then I'm gonna neutralize with media. Sometimes they need a bit of a jolt on the sides. I'm going to do that just to move the cells. Yeah. Yep, so the cells have all come up, maybe 90% of them. So now we're going to neutralize this reaction. I'm going to get the media. It's nice and warm in the water bath. EM. Now we're going to add the same volume of the trypsin, which was 5 mil, so we'll add 5 mil of EM. So now the cells are in this cell suspension. We're going to transfer all of this into a centrifuge tube and put in the centrifuge. gonna label it. Passage 5. All right, there's about 10 mil in here. Balance with the 10 mils. Check that. Yep. 14, yep. Time, yep. Yep. Start. So we'll wait in 7 minutes. We're gonna come and check that the cells have been centrifuged. Okay, so our cell palette should be down there at the bottom. Yep. We have our cells. So now we're going to discard of the supernatant and add fresh media into the pellet. We'll add 5 ml of media. Now we're going to count how many cells are in there. So we're going to count how many cells per 1 mil. So we'll just take 1 mil out of here. Yep, 1 mil. Then we're going to add 100 microliters of trypan blue to 100 microliters of cells, and we're going to use this cell suspension to count in a new centrifuge tube. So we'll use this one. One 100. It up. Yep. And 100 of the trypan blue. Now we're going to use this cell suspension to count. So we'll get the hemocytometer ready. Okay. This is a hemocytometer. We use this to count our cells after adding a dye to distinguish between live and dead cells. The cells that are dead will be blue. The cells that are alive will be transparent. So there's two different tiny little grids split into 4. So when we add our cells in the dye, it will fill this space. Then we'll be able to see the cells, and then we'll have an estimated count on how many cells in each of the 4 parts of the grid. Then we'll find the average, and then we'll calculate how many cells are there in 1 mil. So now we're gonna position this slide on top of the hemocytometer. So that's ready. Then we're gonna add our cells in there. We're going to use that grid on the far end there, and we're going to count how many cells went in there. So this is what the grid looks like. There'll be cells in each of these 4 quadrants. Then we'll add them and find the average. Then we'll calculate using a dilution factor. So now I'm looking at the grid. Yep, you can have a look. Okay, 1, 2, 3, 6, 9, 1, 2, 3, 4, 5, 6, 7, 1, 2, 6, 8. Yep, so now what we're going to do is find the average of all of these. So 6 plus 9 plus 8 plus 7 divided by 4. That's how you find the average. So we calculate it. So there's an average of 7.5 cells in that 200 microliters. So now we're going to multiply this by an equation so that we can find out how many cells per mil. Okay, so the equation is average cells times the dilution factor times 10 to the power of 4. So we know that the dilution factor was 100 microliters of dye in a total of 200 microliters of that. So then our dilution factor is 2. 7.5 times 2 times 10 to the 4. That's the answer. So there's about 150,000 cells per mil. So now we know. So now we can calculate how many cells we need per well. So this is what our 96 well plates look like, and we want to add 5,000 cells per well. To find out how many cells we should add from the calculation before, we use C1V1 equals C2V2, and we put the knowns and we find the unknown. Simple. Cool, so I'm just going to do that now. All right, guys, so we've calculated how many cells we need to fill the 96 well plate. So we've put the cells back into the 37-degree water bath to keep them alive. So now we're gonna take approximately 2.5 ml. We're gonna dilute it in media, then we're gonna dispense evenly into each and every single well. So this is the 96 well plate. We're gonna fill up all 8 columns up to row number 10, okay? So we're gonna make up our cell suspension in this new centrifuge tube. So we're gonna have a look how much we need. So I've just grabbed a 50 ml centrifuge tube, so there'll be enough to make up our cell suspension. So we're gonna add 13.334 ml of media. Then we're gonna add our cells from the water bath into here. Then we're gonna put it into one of these dishes. And then we're gonna put the same volume into each and every single well. First step, add the media. That's 10. That's 13. And we'll use a different pipette to add 334 microliters. And then now we add our cell suspension into there. And we need 2.67 milliliters. So the cells are all in there. So we'll take 2 ml first. Ml. And then 67 microliters. Okay, so our cells are ready in this centrifuge tube. We've got cells and media. Now what we're going to do is we're going to pour it into one of these dishes. It's sterile. And then we're going to use a multi-channel pipette, 8 at a time. And we're going to fill each of the 8 rows. And that's it. Pour. And we need 200 microliters into each. Lock it in. And we can use the same pipette or the same tips. Perfect. that's it. So we put our cells into each of these 96 wells, and we're gonna have a look under the microscope to confirm the cells in each well. Yep. Have a look. Okay guys, so we've plated our 96 well plate with about 5,000 normal colon cells in each and every single well. I've checked under the microscope. So now what we need to do is put in the incubator for 24 hours until we see the cells have adhered to the bottom and they've started growing. Then we can start the experiment.